Welcome to my classroom. So in this lecture number 9, we will discuss about introduction to fermentation and enzyme classifications. Okay. So in lecture number 8, we have seen cell nutrition, in which we have seen the proteins, what are its constituents, what is amino acids, all that have been discussed in lecture number 8. I hope you have completed that lecture. Okay. And we have seen in lecture 8 that various classifications of protein based on their composition, based on the nutritive value, based on their shape and size and their functions. Okay, I will shortly cover these chemical messengers. In that, hormones will be covered right now. Then we will get into the topic. Okay, in human beings, from one place to another place, if you want to pass some information, how we pass that information? If a person is nearby, we will speak to communicate to them, right? So if a king wants to communicate some information to other king, how they communicate? So they communicate through some agents. And even later stages, they even trained doves or any birds are trained, which will remember the path in which it has to travel, and it will come back, right? So that way these even birds like doves are trained to send the messages to from one place to another place. And with the invention of this telegraph machine, people used to send telegrams, right? Any telegram can means earlier days good news or bad news, death news will be communicated through telegrams. So suddenly we have to travel from one place to another place. Then for normal communications, postcards come into picture. And with the invention of computers, all the servers kind of thing in 1980s, we have so many uh, communicating devices came, right? E-communications like Hotmail came into picture, Yahoo Mail came, then Skype calls were came, then Gmail was introduced in 2004, then WhatsApp came into picture from 2009 onwards, then WeChat came. Now we have moved on to get some instant social media messages like uh, SMS and Twitter, WhatsApp and Instagrams are being used, right? So either to increase or decrease. Say even in cricket, coaches will send some information to the captain, right? So they will use the 12th man to pass the information to the captain. Okay, you can take wicket of this batsman by doing this or that, like that, right? So that way even some captain will say that I am going to declare the innings, you accelerate the run rate, like that they will send the information to batsman. Captain will send information to the batsman to the messengers, right? So similarly the cells also will communicate from one cell to another cell or between tissue to another tissue. So the information is passed using some chemical signals. So how do these messages are being sent from one place to another place? Like from cells, we have seen that in the nucleus or DNA, the information is stored, right? So the DNA information stored for function or its repair or maintenance, there some information has to be passed on to the target cell. So how these informations are passed, one of you? Yeah, there are two ways in which these informations are passed. One is through nervous systems. Actually, our nervous system regulates our body with some electrochemical impulses where the signals are carried by the nerves. So, with that, some information is sent and the necessary action is being taken. The other way is like sending some chemical messengers. Okay, so that we will discuss now. So there is like say, some these sources. What we call is that secretion sources or secreting cells will synthesize and release some chemical signals. It's like an organic substance, so that will be synthesized and released. And among the chemicals that among that chemical substance, few substance will reach the receptor or the target cell, and there the information is delivered. That is the way. So we can say that the cells are communicating with each other by means of chemical signals. So these chemical signals is synthesized and released by the secreting cell, and it is transported to a target cell where it is recognized and causes the target cell to alter its activity in some way. So these chemical signals can be in many ways. One of, for example, pheromones, hormones, local hormones, neurotransmitters, cell surface molecules, or intercellular messengers. So these are the chemical ways in which the chemical signal can be done. So these five major signaling systems in the cells all begin with a chemical signal binding to a receptor. First thing is the receptor this is a chemical signal, the chemical substance should go and get bind to the receptor. Okay, so one of these is used by the steroid hormones that bind to intracellular receptors, whereas the other four pathways begin with the hydrophilic chemical signals binding to plasma membrane receptors on the cell surface. Okay, so the among the five major intracellular signaling systems like steroid hormone action, then we have ion channel linked receptors, you can refer to this image, we have camp pathway, and we have phosphinocytide pathway, then we have tyrosine kinase linked receptors. Okay, so there are two ways by which the communication is carried out, that is the information stored in the DNA to how to reproduce, how to grow, how to repair, all that information is stored, right? So that information is passed on or based on the environment, a decision is made and the decision is sent to the particular location to carry out particular operation, right? So among these five, first one I will discuss. The first one is steroid hormone action, where the steroid hormones diffuse across the plasma membrane of the target cell and bind reversibly to specific steroid hormone receptors in the cytosol. So the steroid receptor complex then undergoes a conformational change which allows it to bind to DNA to switch on the transcription of early response genes which produce signals to activate other genes. Okay, so that way you can see this figure. So all the cells have evolved a chemical signaling pathway that enables them to communicate over a long pheromones or hormones. The intermediate, which is done using local hormones, are very short information like neurotransmitters or cell surface molecules or intercellular messenger distances. So the distance may vary, it may be a long distance or a short distance. For example, these different chemical signals that span a range of distance starting from a small distance of nanometer range up to some kilometers also. Okay, within the cell I am talking. So these target cells responds to these chemical cells using a limited repertoire of 5 intracellular signaling pathways that decodes the incoming signals by processing them into forms that can be recognized by the response elements that bring about the change in activity. Okay, so that response activity, you look at the figure, so it can be secretion, it can be a metabolism, it can be contraction, or it can be cell growth, or it can be excitability. Okay, so that I wanted to complete in the previous lecture. And in this lecture, if you start with what is the basic principles of fermentation, I hope you might have read it earlier also. So just we will rewind with what is the definition of fermentation. Okay, so to start with, if you look at these three things shown here, you have seen some beverages are there like beer, yogurt, and you have a sprinter. So what the sprinter means, the people who will complete a short distance in quick time, like Usain Bolt, who has won 8 Olympic medals, Carl Lewis, who has won 6 medals. So they are called sprinters. So what is common between these three people? So those who are attending this class, only you people, refer to this chat box, fill this form, fill your responses. Yeah, good. My job is made simple, so I'll just proceed. You can fill that form for attendance. Okay. So what is common in all of this? Yes, most of you have made it clearly that all of them are involving fermentation. So in fermentation means what is happening in case of this left is alcohol fermentation is done to make beer. In the center, lactic acid fermentation is taking place for making yogurt. Whereas in case of the human being or animals like human beings, what we are doing is whenever there is a lack of oxygen, when we run short of oxygen in our body, a kind of lactic acid fermentation is taking place. Okay, so that is the commonality between those three things. Good, thanks for your responses, those who have made. So just formally make that definition, what is fermentation and all. Okay, so if Louis Pasteur, according to Louis Pasteur, it is far from being a lifeless phenomenon, it's a living process. So there are several examples. Actually, how that name came, before that we will see what is aerobic cellular respiration. So that is an example for that is like respiration that is taking place in human beings. And we have some anaerobic cellular respiration. For example, lactic acid is produced in our muscles. Okay, what happens in that, we will see that one. Okay, so before that we have seen that what is glycolysis. So actually during glycolysis, that glucose is converted into pyruvate, along with it will produce some 2 ATP molecules, which is a universal mediator, adenosine triphosphate, and it produces NADH, that is nicotinamide adenine dinucleotide plus oxygen. So that, and also they will produce some 3 carbon molecule pyruvic acids. Okay, so that way this is the reaction that is taking place. And despite the fact that this energy from this glucose was used to make this ATP, NADH and the third product, they still have some lot of chemical energy rest in it. Okay, so if you look at what is ATP, it is one of the important chemical to all the lives. It stores and releases the energy for many cellular processes. It is found in this mitochondria. And NADH stands for this nicotinamide adenine dinucleotide along with hydrogen. This chemical occurs naturally in the body and plays an important role in chemical process that generates energy. And still, if you look at pyruvic acid, is a 2-oxo-monocarboxylic acid, that is the 2-keto derivative of propionic acid. Again, it is a metabolite obtained during the glycolysis. So what is aerobic cellular respiration? It means that in presence of oxygen. The aerobic word means that with air. So this type of respiration needs oxygen for it to occur. That's why it is called as aerobic respiration. Example for that one is glucose with oxygen will form carbon dioxide plus water plus energy. Okay, so actually this reaction follows 3 stages. One is glycolysis stage, citric acid cycle, electron transport chain. So that in biochemistry courses you could see that detailed reaction that is taking place. But overall, if we summarize, what happens is different stages in this aerobic cellular respiration. So during this first step, glycolysis, a net of 2 molecules of ATP or chemical energy is produced. And what is glycolysis? It is a cytoplasmic pathway which breaks down the glucose into 2 to 3 carbon compounds and generate energy. And the citric acid cycle also will produce 2 molecules of ATP. And during this electron transport chain, it will produce around 28 molecules of ATP. So that way totally you have 36 ATP being roughly produced during this aerobic respirations. Okay, so oxygen is mainly used in this aerobic respiration as the final electron receptor. Okay, so how to define then this aerobic respiration? It is a set of metabolic reactions that take place in the presence of oxygen occurring in a cell to convert a chemical energy into ATPs. How many ATPs? Around 36 ATPs are formed. Okay, so but what will happen when there is no oxygen in it? When there is no oxygen, what will happen to the same reaction? Will it proceed? How many steps? We have seen that 3 cycles are involved. What we do when you are not able to breathe? We suffocate, right? We become even unconscious. So what happens during that time is, in absence of that, we will not have all the 3 steps being done. Okay, only the first step will take place. Okay, that is glycolysis only will take place in the anaerobic cellular respiration. So that's the thing. There will not be further thing taking place. For example, glucose becoming glycolysis, then there can be aerobic respiration or anaerobic respiration. So if you clearly look here, in presence of oxygen, what they do is they produce around 30 to 32 ATPs. Okay, whereas if in absence of oxygen, what happens is this glucose is getting converted into ethanol or lactic acid fermentation. So that way, in that case, only around 2 ATPs are produced. So that is the difference between aerobic and anaerobic things. So if you want to summarize actually if you can detailed thing we discussed earlier also. So we'll just compare them with respect to definition and the overall reaction taking place. So what are the end products? If you look at here, these end products are of aerobic respiration are CO2, water and energy. Whereas in case of anaerobic respiration, the end products are some acids can be produced, some alcohols or gases or energy. Along with energy, this can be produced. Okay, so if you look at anaerobic respiration, that will only occur in the cytoplasm of a cell, whereas in case of aerobic respiration after glycolysis occur in all these mitochondria, eukaryotes, cytoplasm of prokaryotes also. Okay, we have in detail we have seen what is the difference between eukaryotic cells and prokaryotic cells. Okay, so what type of gas is being exchanged in these two cases? In case of aerobic, there is an exchange of gases during aerobic respiration. Oxygen is absorbed, CO2 is released. That's why CO2 is there in the end product. Whereas in case of anaerobic, the exchange of gases does not take place during this anaerobic respiration. However, there may be some some amount of gases like sulfur and nitrogen gases are released by certain specific organisms. Okay, so mainly it is in presence of oxygen or in absence of oxygen. And with respect to the energy produced, around 38 ATPs are produced during anaerobic respiration, and only 2 ATPs are formed during anaerobic respirations. Here the reactants are carbohydrates and oxygen are the prerequisite for aerobic respirations, whereas some electron acceptors like sulfur and nitrogen are required for along with carbohydrates for anaerobic respirations. Okay, so which one is longer? If you ask, aerobic is comparatively longer than the anaerobic respiration. Okay, so where does it occur, aerobic respirations? Aerobic respirations occurs in most of the higher organisms like plant and animals, whereas anaerobic respiration occurs in primitive prokaryotes. Okay, so anaerobic respiration also takes place in the muscle cells in human being during extreme movement like sprinters, what they are doing. Okay, so that way in prokaryotes anaerobic respiration is done. Okay, so if you look at the definition of this word fermentation, how it got this name, if Latin word in a Latin word fervere, it means to boil. Okay, so fermentation is actually another type of anaerobic respiration that takes place in anaerobic organisms like yeast. Mainly yeast only we will be using here. Okay, so when any carbohydrate rich substrates are bottled, that means we call it a substrate, right? So when carbohydrate rich substances are bottled with yeast, ensuring minimum oxygen content in the bottle, so this yeast will undergo anaerobic respiration. So as a result, what happens? This fermentation will take place, and these carbohydrates are converted into alcohols like ethanol. Okay, so this alcohol produced in the bottle is, however, toxic to the yeast. So what happens? Then yeast will commit suicide. That yeast will start dying as the alcohol concentration increases. Okay, so only about 30% of alcohol can be brewed with yeast, while higher concentrations are obtained through distillation process. Okay, as in lactic acid fermentation, fermentation also results in just 2 ATPs as energy. So the overall reaction is given here. So along with CO2 and energy, ethanol is formed. So we can say that if you want to define what is fermentation, it is a process by which the living cell is able to obtain energy through the breakdown of glucose or other simple sugar molecules without, means in absence of oxygen. So who found this thing first? If you look at history, Louis Pasteur in the 19th century first coined this term called as fermentation, because he observed that these changes can be brought out using the yeast and other microorganisms in the absence of air. He also recognized that it's not that only with this kind of yeast we can only produce alcohol or carbon dioxide. There can be many other products if we are able to use them wisely. Okay, so in terms of biochemistry, we can say that it is a metabolic process in which what is metabolism? It is a sequence of several biochemical reactions, right? So that's why we are concerned about it. So fermentation is the metabolic process in which carbohydrates and related compounds are oxidized with the release of energy in the absence of external electron acceptors under anaerobic conditions. In broader sense, if you look at any dictionary, Oxford dictionary, and see what is fermentation, they simply say it is an enzymatically controlled transformation of an organic compound. Okay, so there are wide applications of this fermentation is there. So it's a means to harvest energy from the food. The food waste is there, or food from the food also energy needs to be taken. So that is also involves fermentation, and it is employed by the simplest single-celled organisms, where we can convert this biomass into or municipal solid waste, kind of any waste, any biomass waste can be converted into some useful energy or other things, okay, by use of some microorganisms, okay. Also they can make use of this yeast, which can turn this grape juice into wine, which we can even use to produce ethanol or CO2, all that, okay. And in absence of oxygen, that process is called anaerobic one. So we have several other applications. We will discuss fermentation in detail later, but if you look at some of the wide range of applications, it can be like used for to make some microbial cells like yeast. It can be used to make microbial enzymes like glucose isomerase. We are going to discuss enzyme after a while. So these microbial metabolites like penicillin can be produced, food products like cheese, yogurt, vinegar, and to produce some vitamins like B12, riboflavin, all that. Okay, so they have several products which can be classified as food products. That's why in food industries, these biochemical engineering or these fundamentals are very important. Fermentation, understanding how the fermentation is done, all that, okay. And with respect to production of industrial chemical solvents like acetone, butanol, ethanol, enzymes and amino acids, or for even specialty chemicals for medical applications like pharmaceutical and vitamins can be produced, okay. So even you have these products which can be used in end products, like you see that in our daily life, all these things we have been using. So in our end daily life products, these fermentation products are involved, okay. And we are going to discuss the reactors in the third chapter. Just now to start with, there are two type of commercially available reactors are being used. One is solid state fermenter and another one is submerged fermenter. It is one like we conduct the experiment on the surface of something, okay. So that is the one way. Another way is in the submerged fashion, just like in fermenter reactions. So we have two type. What is being done in these two type is, one is solid state, another one is submersion type. So the microorganism cultivated on the surface of a liquid or a solid substrate, and whereas in case of submersion techniques, the microorganism grow in a liquid medium. Okay, so this solid state is little bit complicated in comparison to the submersion techniques. So that way, this solid state is rarely used in the industries. Okay, some of the examples for solid state techniques products are mushroom, bread, cocoa, all these are produced. Whereas using submersion techniques, so biomass, protein, antibiotics, enzymes, sewage treatment are carried out using mostly by these submersion techniques. Okay. So some of the industrially important products for the fermentation are, you can see that ethanol, which microorganism used and what is the end use. This table, you can see these things are being used. Okay, so ethanol using Saccharomyces cerevisiae, we get several industrial solvents and beverages, and you can produce glycerol, you can produce lactic acid, you can produce acetone and butanol, alpha amylase. So all these can be used using respective microorganism. It's not the only one microorganism, it is widely being used or well established to make use of this particular microorganism. Other organisms, if you can able to identify, we can use that one. As I said, we can even isolate from a given source and then make use of that particular microorganism for any reaction. Okay, so now we will get into these enzymes. What is enzymes? Enzymes are actually catalysts of life. You can say that they are the catalysts of life. So what is the role of a catalyst? We have seen in CRE course also, just like a relation between a teacher and a student. If you people start reading from a textbook, it's not for the bookworms, okay. So in general, if we want to read ourselves some 300 pages, we find it difficult, or we get tired, or we get something not interesting also. So that way, if we are able to learn it through our friends who became a teacher, right, for overnight study, we used to do the same. When there were no internal marks, we'll only study for end exams. Means what we'll do, we just during the study holidays, one fellow will study all the subject, 10 students around will be sitting around him, and he'll start explaining that. So when you are learn through our somebody who can able to explain us the way we want to understand, or we will able to understand, so that will be acting like, he is acting like a catalyst, right, in the learning process. So for a learning process, a teacher or a friend is a catalyst. Okay, so similar to that, in the living organisms, to carry out the activities in more effective ways, these enzymes are acting like catalyst for life. Okay, So actually these enzymes are proteins in nature, and there is a non-protein enzyme also, except one. Most of the enzymes are protein in nature. Okay, so what there is one thing called as ribozymes, that enzyme alone will not have protein content. It will be rich in group of ribonucleic acids, so that so that activity, whatever function that catalytic enzyme activity is due to that RNA present in it. Okay, so except that one ribozymes, most of the enzymes are protein in nature. Okay, so if you look at the history of these enzymes, so in 1883, so I'll repeat, if you look at the history of this enzyme in 1883, Buchner identified this enzyme called as zymase. It's an enzyme system actually, it's not single enzyme. Okay, so he isolated this enzyme system from the cell-free extract of yeast. From the extract of the yeast, he isolated out the zymase. Later it was found to be a group of enzyme. That's why we call it as enzyme system instead of enzyme. Okay, so which could convert the sugar into alcohol. So this is the first person in 1883 he did this one. Okay, and in 1926 James Summer, in 1926 James Summers isolated urease, which is isolated from some jack bean. Okay, so this is what historically first, before that we know that in chemical reaction somebody coined the word catalysis. Okay, in 1836 or in 1878 Kunhe is the first person. Okay, in 1878 Kunhe coined this word enzyme, which means in the yeast, means in the yeast. It is first isolated from that, right? Okay, so that was the first thing. In 1878 Kunhe could able to isolate this enzyme in the yeast, and in 1883 he isolated an enzyme system called zymase, which can be utilized too. So that way here only isolated, here he identified the application that sugar can be converted into alcohol. In 1826 James Summer isolated the urease from some jack bean. Okay, and this can be actually, this is in this only he first identified that it is a protein. So till then they didn't know that the enzyme is a protein. So he identified that this urease enzyme is made up of protein, and the naming of that came here only into picture. Okay, so if you look at the nomenclature of this enzyme, how people used to name these enzymes. So for example, if you look at initially whoever discovered it, based on their name these enzymes were named. Okay, for example like pepsin, like trypsin, or chymotrypsin. So these are some of the enzymes which were named after the discoverers. So based on the discoverers, some of earlier days they used to name the enzymes. So then came a case where simply a word at the end as a suffix ase is used. Ase was added to the substrate. Ase at the end of the substrate. That means you have the substrate from which this is made. So it means the substrate which it is converting into some useful product. So then they added to which substrate it can convert into product. To that they added the name ase. For example, lipase. What does that mean? Here lipid is the substrate which it can convert lipid into another product. So they called it as lipase. And similar to that, if it can nucleic acid, if it is converting it, then an enzyme called nuclease. So this fashion people started naming, adding ase along with the substrate at the end of the substrate, not prefix, it's a suffix. Okay, So they started adding a suffix ase to the substrate, like so lipid, if it can convert the lipid, it is called as lipase. From nucleic acid, it is called as nuclease. If it can use lactose, then what would be the name? One of you? Lactase. Okay, so that way people started naming the enzymes this fashion. Okay, so all these enzymes can be classified into two broader cases. Okay, two broader classifications are what are two broader classifications of this one? Okay, so one is intracellular enzymes. Okay, so these intracellular enzymes, they are functional within the cells where they are synthesized. We have seen that within the cell these enzymes are produced. Okay, so cell inside the cell wall. So inside the cell these enzymes are produced and they are operated. They are functional, means it will do its role only within the cell. Then they are called as intracellular enzymes. So they are functional within the cell in which it is produced, cell in which it is synthesized. Okay, so then what is extracellular enzymes? You can take it out. That means you can say what is that we can do. We have seen several cell disruption techniques, right? In downstream processing, in cell lysis, lecture number 6, we have seen what are the various methods in which we can take out the products present inside this. So that way you can take it out and make use of them, like whatever product like acids are being produced, right? So that way they are called as extracellular enzyme, which can be taken out and used. So they are active. So these extracellular enzymes are active outside the cell. We can take it out and make use of them as the catalyst. Okay, outside the cell it is synthesized, in which it is synthesized. Okay, so all the digestive enzymes belong to this category of extracellular enzymes. So that example for extracellular enzymes are all digestive enzymes. So that's why several tablets are given now, right? If you are having digestive problem, people will take these enzyme tablets. Okay, so they will help in the digestion. Okay, and what happened then is, so there were a lot of ambiguity, or a lot of different names are given, right? So in 1961 to 1964, so this International Union of Biochemistry, so all this coming under biochemistry, so this International Union of Biochemistry, what they did is they formed a committee and they told them there are so many enzymes are being developed and named it, names are there. So we want to make a uniform pattern and classify them, and we want to easily educate people, or people want to use a particular enzyme, how to make use of it, how to classify them. So they formed this committee. Okay, so what they did is this committee has derived with basic things. Okay, for that you have to understand, right? I hope in my master answer class to you I might have explained. So how this one of the person who was instrumental in visiting several industries like chemical industries, pharmacy, means all the chemical industries, pharmaceutical industries, cement industries, polymer industries, all places he visited, and he identified that here these unit operations are done. Several unit operations and unit processes are common, only the scale in which they are used are different, right? So like that he would be able to identify. Then only the concept of chemical engineer came into picture, because he was able to classify them into unit operations and unit processes, and we are able to say the principle is same. Only the small scale grinder is small scale in one industry, it is big scale in industry. Distillation is small unit in one industry, it's a big unit in a petrochemical industry, like that. So we are able to give birth to this chemical engineering, right? So similar to that, these people also are able to identify them. Okay, so and they classified them into 6 major classes. Okay, so which comes under this OTHLIL. So to memorize this, they asked us to keep this word OTHLIL. So there are 6 major classification. Each classification represents the general type of the reactions brought about by these enzymes. So it is like based on the name. So whatever we have classification made here earlier, so in all these places we don't know what type of reaction they will catalyze. So what is the nature, what type of reaction it is taking place, right? So the metabolism is not explained in the name, because looking at the name we are not able to tell what type of thing it is going on, right? So maybe here it is useful in identifying what substrate it will work on. So if I want to convert a lipid, the substrate, then lipase can be used. To nucleic acid substrate, nuclease can be used, like that. So some information is given. Here also it only gives the information on the discoverer. Okay, so but they are not giving us the classification, means the nature or the role that is going to play, what type of reaction they can catalyze, they undergo, all that is not given in this one, right? So that what based on that these people classified them into 6 major categories. One is when they are able to catalyze the oxidation and reduction reactions. So oxidation or reduction reactions. So that's like AH2 plus B is there. So they are able to make it into A plus BH2. So this type of reactions, if they are able to do, then they are classified as oxidoreductases. Up to this oxidation reduction, aces refer to enzymes. Okay, so this oxidoreductases refers to the enzymes which will undergo oxidation or reduction reactions. Okay, so that is one classification. For example, alcohol dehydrogenases. So this is the enzyme example for coming under the classification oxidoreductases or L&D amino acid oxidases. So these are some of the examples enzymes for undergoing this type of reaction coming under this classification. Okay, so then there can be another type of reaction called as functional group transfer. So for example, A is containing some X functional group and B is there, so they may be converted into A plus BX. So such type of functional group transformation, transfer of functional groups. Okay, so this type of reaction, if it can catalyze the functional group transformation reactions, so they are called as transferases, as per the name, transferases. So when you say transferases, so they are the enzymes which will undergo the reaction type where the functional group is transferred. So examples for that are some enzymes which will undergo these transferases are hexokinases, or you can say transmethylases, and then you have phosphorylases. So these are some of the enzymes which undergo this functional group transfer reaction: hexokinase, transmethylase, phosphorylase. Okay, so these 3 are examples for the enzymes which undergo transfer of functional group. Okay, so then the third one is hydrolysis. Hydrolysis reactions, that means if you have AB plus H2O, it will form AH plus BOH. So this is hydrolysis reaction. So when enzyme which can catalyze the hydrolysis reaction, they are called hydrolysis. Okay, so example for this type of enzymes are lipases, trypsin, and urease. So these 3 enzymes are examples for the enzyme which will catalyze hydrolysis reactions. Okay. So then we have addition or removal of water type of one is there. Addition or removal of water. So what is that? We have AX plus BY is there. So what it will form? It will form AB plus XY. Okay. So this type of reactions, if we can catalyze, they are called as lyases. Okay. So example for this type of enzymes are fumarase and histidase. So this will catalyze the reactions involving addition or removal of water. Okay. Then we have isomerase. We know isomerization reaction, right? So isomerization reactions. So what is isomerization reaction? So this A will change into another form. Okay. So such type of enzymes are called as isomerases. Okay. So these enzymes are specialized in isomerization reactions. So example for these are retinol isomerase and phosphoexose isomerase. So this is one type of classification. Okay. There is another final and last type of this classification they have given is the enzymes that will catalyze some of the synthetic reactions. So actually it will do involve in some binding reactions. Okay. So the meaning of the word ligate actually stands for to bind. So this was some binding reactions if it is catalyzing. So they are called as ligases. So remember this is another type. Sixth type is ligases, which will help in the binding reactions of some synthetic reactions. Okay. So these are the classifications given by them.
So in this table, you have in the tabular form, you can list out these classes and what type of chemical reaction it is catalyzing and some examples for the enzymes under that particular classification.
And beyond that, they have classified the enzyme commission. So they formed actually some EC number they are given. It is called enzyme commission number. Okay, so that EC number is given. It is like 4-digit they have given. It's like book chapter. Some books, no, they will have one chapter 1.1 refers to the subheading, 1.1.1 refers to subheading, 1.1.1.1 refers to subheading, right? So that way they are given a 4-digit one. So where the first one will give the class, first one is class, the second one is subclass, third one is subclass, and fourth one is the individual enzyme name number. So that way individual enzyme. So that way they are given a 4-digit EC number for this thing. Okay, so that's what they have proposed. But however, people have difficulty in following this 4-digit number EC number given by them. So still people are using the thing. Okay, so this International Union of Biochemistry, our names are more specific. These names are more specific because by looking at, if you know this number and remember what the type of class it belongs to, it's like our roll number in our school, college also, right? We have first 2 digits and 3 digits. What does it stand for? One of you in our roll numbers? What does this first 2 digits and third digit stand for in your roll number, student roll number? One of you? For example, a student is starting with 106,101, or we have 186,101. Okay, what is the difference? We have a student 206,128, and we have 205,128. So how are you classifying these people? Between these two, what is the change? And between these two, what is the change? One of you. Anybody there? Or all left? Yeah, Vaishnavi. Himavati. Are you there?
Yes, sir.
Yeah, tell me what is the difference between these roll numbers? 196101 and 186101. You know that?
Year of joining.
Yeah, first two refers to year of joining. Second one, okay, between these two students, you tell me, you have 2020 refers to these people joined in 2020. Okay, then what is the difference between these 6 and 5?
Branches.
Yeah, very good. This person belongs to chemical engineering. What about this 5? Which department? We have ranked this third digit belongs to 1, 2, 3, 4, 5, 6 as per the seniority by which the department is established on our institute. So ours was the 6th department, chemical engineering. So we have this 6 here, right? So similar to that, they have also done the classification, which is you will be able to identify the some data, right? If I know this number 1, 2, 3, 4 is there, if I know this one refers to a particular class, this belongs to a particular subclass, this belongs to a particular thing, so I am able to easily identify that one. That is the objective with which they have given it. But still people have difficulty in mugging and remembering all these things, right? So that way, so but however this is very specific, right? I am able to identify a student, locate. I am able to locate a student, which batch he is, which branch, which year he joined, which branch he joined, which section. One means first section, 201 means second section, right? So like that we are able to classify. Similarly, these IUB names are very specific and they are very clear also. Okay, so they are very specific, means they are very clear. So, but they are not widely used because people still used to use the same old name. So that's why still people are sometimes using only trivial names. So whatever I have discussed earlier based on discoverer and based on adding A's at the end, so they are called trivial names. So still people use this trivial name, and if required some places they use trivial name plus EC number. So that way in books, if you want to buy the enzyme for particular reaction, you should able to mention both. Okay, trivial name as well as this what is the EC number, like that you should able to give. Okay, and how we will choose a particular enzyme? Okay, so because we should know that enzyme activity is an important thing, right? So enzyme activity, what is enzyme activity? I'll take 10 minutes and stop. Okay, we will discuss this and stop. What is enzyme activity? We are slowly getting into the core of the course. Okay, whatever we discussed till earlier lectures are only basics which we should know in proceeding in this course. So enzymes are never expressed in terms of their concentration, like in terms of micrograms or milligrams per liter kind of thing. Okay, so they are not expressed in terms of milligrams or micrograms per liter. How? But these enzymes, whenever we say enzyme, we talk only in terms of its activity. So we should know what is that enzyme activity. So enzymes are never expressed in terms of the concentration, but they are expressed in terms of the enzyme activity. Okay, so there are various methods are there, and various techniques are available, and different units are used to express what is the unit of this enzyme activity. So we have different units available for it. Okay, so one of the unit is Ka unit, which refers to King Armstrong unit. Another unit is called Somogy unit, and another unit is Reitman Frankel units. Okay, so these three, and there is one more unit called as spectrophotometric units. So like that, there are different units for this enzyme activity is having, so which we will use, and in this mainly there is a unit called CATAL unit, which is, or short form it is CAT. We'll say enzyme activity is 100 CAT, like that, no? So this CAT or CATAL, in full it is CATAL. So this is one of the unit. So this represents how many moles of, what is the concentration of 1 mole of substrate per second. Okay, so it is the, so 1 mole of substrate is converted. So we can say conversion of. Okay, so the conversion of 1 mole of substrate per second is taking place. It is called 1 cat. So 1 cat represents the conversion of 1 mole of substrate per second. So this can be subunits, because there can be, it can be expressed as milli, say it's like meter, mean there can be millimeter and micrometer. So that way you can have like 1 millicat, or you can have even 1 microcat. Okay, so accordingly you have the conversion factor, right, in the scale of -3, -6. Okay, so that way this is one way of using the unit for this catalyte. So it represents the conversion of 1 mole of substrate per second. Okay, so then later stage people came up with some international unit system. So international SI unit system came into picture later stage. So then people wanted to express it in terms of universally we want to have a common unit. So we started defining one term called as international unit. What is IU? What is 1 IU? We say similar to it, that little change is there. Okay, so if 1 IU represents, 1 IU represents the catalytic conversion of, that is the enzyme, it is the amount of enzyme activity that catalyzes the conversion of 1 micromole. So in SI unit system it is 1 micromole of substrate conversion. Per, here it is second, right? Here it is per minute. So 1 IU is equal to, that is 1 international unit is the amount of enzyme activity that catalyzes the conversion of 1 micromole of substrate per minute. Okay, now we can easily, so we can easily observe that they are similar units only. One is CATAL, which is 1 mole of substrate per second conversion, it is CATAL. If it is 1 micromole per minute, it is 1 IU. So I ask you to find out what is the relationship between 1 IU and 1 CATAL. So find what is the unit conversion. Please carry out. Having notebook with you. How they are converted into each other. What is the conversion factor? You know that 1 micrometer is 10 power minus 6 meter, right? Similar to that, here also you will be able to convert it. And tell me 1 IU is equal to how much micromole per second? When I say that, it is equal to how much microcats? So what is the value? Put it in the chat box. Do the conversion and paste it in the chat box. What is that simple definition? As per definition, 1 IU, 1 IU is it is the catalytic activity that can convert 1 micromole of substrate per minute. Here it is conversion of 1 mole substrate per second. So that time we should convert the unit of second to minute. Then micro macro thing you have to take care. There is a micromole, there is a mole. And if you ask me what are the advantage of these enzymes over the other chemical catalysts is, so these biocatalysts take very less time. Okay, so this biocatalysis is gradually slowly taking over from the chemical catalysis in many industrial operation, because we want to reduce the pollution. We are moving towards green technologies, and that's why these people move towards biochemical processes or metabolisms, whatever involved. So that way we make use of those microorganisms, or this mainly these enzymes are widely used to this thing. Okay, so that is the advantage. And also it is going to be environmental friendly, and they are biodegradable, and they are more efficient in terms of cost, all that. Okay, so that way these are very important. So if you look at the applications, so there are several industries which make use of these enzymes, and they are mainly used as therapeutic agents. So for example, enzymes are used in the therapeutic applications, like the streptokinase, which is used to blood clotting issue is there. We want to remove the blood clots. So in that case, these enzymes called as streptokinase is used. And another second usage is in analytical laboratories, right? So we have seen that in blood test, you go to some lab to test blood sugar. Nowadays all digital instruments have come, we are able to order it in online and do, but earlier days we depended upon these laboratories which will do this thing, right? So in laboratories, these analytical reagents, they are used as these kind enzymes are used as analytical agents, where they are used to measure the quantity of the substrate or the drugs activity, or the even in urine we are able to identify how much is the glucose content in urine, like kind of things. Okay, so that's why this estimation of plasma glucose using the one of the enzyme called as glucose oxidase, so we are able to do, or even the peroxidase methods are even used. And also these immobilized enzymes are being used in the industries, and these biocatalysts are mainly used for industrial as well as medical applications. Okay, so what is immobilization means? They are immobilized by binding them to a solid substrate, which becomes an insoluble matrix, which will not affect the, we should do immobilize it such a way that it will not its activity is not affected. Okay, or even its stability should not be affected. So that way these are embedded onto the some surface. For example, bedded gels or cyanogen bromide activated sulfurase are commonly used to immobilize this enzyme. So in the form of immobilized enzymes, they are widely used in several industries. So for example, these glucose oxidase and peroxidase, they are immobilized and coated on a strip of a paper. So when these two enzymes, what is that, glucose oxidase and peroxidase, they are immobilized and coated on a strip of paper. So what I mean is they are used in the clinical laboratory. We are coating two, one A and B. What I am saying is A is glucose oxidase, so B is peroxidase. So glucose oxidase and peroxidase are coated on a paper, and you take a sample of urine, and when this is dipped into this, because our urine will contain the different level of sugar based on our age and the food practice that we follow, right? So we have this one. So when this strip is dipped into it, it is going to change color from colorless to blue color. Okay, so initially there will be colorless, and it will become blue color. So what happens, that reaction, what is the reaction taking place? Okay, so what happens here is we have glucose present in the urine sample, and first thing is this oxidase is there, right? Glucose oxidase. So it will convert this glucose into gluconic acid and H2O2. Okay, so then further this H2O2 is taken care by the second enzyme, peroxidase. Okay, so this will convert this into water. So these two reactions take place when we take the urine sample and put this strip made up of A and B into it. Okay, what happens here, actually I told that it will turn the color from colorless to blue, right? So what is happening there? Here we have Oledine is converted into oxidized Oledine. Okay, so that is why this color, this oxidized Oledine will be blue in color, right? So that way, so this, it is like a semi qualitative amount. Okay, semi quantitative, we are exactly don't know what is the sugar content, but 50 we are able to know that actually we can make a calibration chart, right, with different levels of glucose, concentration of glucose. We can, if you know, with the known sample we can make a different concentration of glucose solution, and then we can know what is the concentration, we can make activity the thing, right? So that sugar level versus concentration plot is there, we can able to use it to quantify it. But when we are doing this kind of strip method, it is like semi quantitative method. So the intensity of this blue color, right, so how dark blue it means that the intensity of this blue color is a function of, the intensity of this color is a function of the amount of sugar present in the urine, right? So that way, if it is very light color, we can say that okay, sugar level is low. When it is dark blue, we can say that the sugar content in your urine is more. So that means you may have, your sugar level may be high, right? So this strip method is used as a semi-quantitative estimation of glucose in our urine samples. There is another method called Oledine method for estimation of blood sugar, blood glucose. So this is one of the oldest method to measure the blood glucose on estimation. Nowadays it is not used because this particular chemical is believed to be carcinogenic in nature, and that's why nowadays enzymatic methods are used. In this method, these proteins are first precipitated by trichloroacetic acids. The glucose present in the protein-free filtrate reacts with toluidine in a hot acidic medium to form a stable green colored complex, namely N-glycosamine. The presence of thiourea stabilizes the toluidine reaction. The intensity of the color developed is measured photometrically at 630 nanometers, which is directly proportional to the concentration of glucose present in the fluid. Okay, so this method is widely used. The procedure is shown in this figure also. You can go through it. So this is one of the example. With that, we would like to stop. Okay, so we will continue in the next lecture. Thank you so much for joining my class. Those who have questions can stay back, others can leave.